Hello guys, this is Dr. Ravi Sani. So today we are going to discuss about the regulation of blood pressure. So in case of regulation of blood pressure, basically we have two broad mechanisms. The first one is the short-term regulation and the second one is the long-term regulation. The short-term regulation basically it is called neural or it is done by the nervous system. In case of long-term regulation, it is mainly done by the hormones or the renal system. These two are the broad mechanisms through which regulation of blood pressure could be done. Now come to the short-term regulation. The first mechanism in short-term regulation is the neural mechanism. What are the various nervous system parts are involved in short-term regulation? So in neural mechanism, the first one is the autonomic regulation. Through the autonomic nervous system, there is a control of blood pressure in which we have sympathetic control and the parasympathetic control. The second one is the medullary control, hypothalamic control, cortical control, and there are some reflex regulations. There are some reflex mechanisms through which short-term regulation could be done. The important ones are the baroreceptor reflex, chemoreceptor reflex, Cushing reflex, somatosympathetic reflex, atrial stretch reflex, ventricular and pulmonary stretch reflexes, non-physiological chemoreflexes, which are also named as coronary and pulmonary chemoreflexes. And in the last, there is a vein bridge reflex. So these are the various reflex mechanisms through which blood pressure controlled in a very short span, a short duration. And the other parts of the nervous system which involved in short-term regulation are the cortical control, hypothalamic, medullary, and autonomic nervous system, which have two components, sympathetic and parasympathetic. There are some other mechanisms which are responsible for the short-term regulation. The first one is the vascular mechanism, like capillary fluid shift, stress relaxation. These are some mechanisms through which blood pressure could be controlled in a very short duration. The third one is the hormonal mechanism, various hormones or chemical compounds which are responsible for the short-term regulation. The important ones are the catecholamines, renin-angiotensin system, anti-diuretic hormone, atrial natriuretic peptide, kinin-kallikrein system, histamines, endothelins, EDRS, and adrenomedullin. These are some chemical compounds which are responsible for the short-term regulation of blood pressure. So this picture is giving us a very brief idea about the various mechanisms through which short-term regulation of blood pressure is done. Now come to the neural mechanisms. So areas of the brain that play important roles in the nervous regulation of the circulation. So various areas or various cardiovascular centers which are mainly located in the medulla. These centers primarily control the autonomic effect on heart and blood vessels. So these cardiovascular centers which are present in medulla basically control blood pressure through the autonomic effect on the heart and blood vessels. In this image, we are having these centers, the vasomotor center, a vasodilator center, vasoconstrictor area or the vasodilator area, and the vasomotor center. These three are very important in regulation of blood pressure. This one is responsible for the vasodilation. It does it is responsible for the vasoconstriction, and vasomotor center uh, collects signals uh, through the various receptors. So these medullary cardiovascular centers broadly divided into two. The first one is the vasomotor center. The second one is the cardio inhibitory center. In case of vasomotor center, we have three specific areas. The first one is the vasoconstrictor area, vasodilator area, and sensory areas. This is the location of these areas. It is responsible for vasoconstriction. It is responsible for vasodilation, and sensory area collects inputs from various receptors. In case of cardio inhibitory center, we have nucleus tractus solitarius, nucleus ambiguus, and the dorsal motor nucleus of the vagus. So these are the cardio inhibitory center. Stimulation of these areas results into bradycardia or decreased cardiac output. So basically, this bradycardia or decreased cardiac output mainly done by these two mechanisms. The first one is through the vagus nerve, which basically originates from the nucleus tractus solitarius, and on stimulation, it inhibits heart functions. The second mechanisms are the various local inhibitory interneurons, which inhibits vasomotor center. Stimulation of vasomotor center causes vasoconstriction and cardio acceleration, and inhibition of vasomotor center causes vasodilation and cardio inhibition. So vasomotor center is a very important part in the regulation of short-term regulation. Basically, when there is a stimulation of vasomotor center, it causes vasoconstriction and it causes cardio acceleration. It increases heart rates and cardiac outputs. But in case of inhibition, there is a vice versa results like vasodilation and cardio inhibition that decreases blood pressure. Sensory areas receives sensory impulses via glossopharyngeal and vagus nerve, which are the mixed nerves, and these nerves basically receive impulses through the baroreceptors, which are the pressure sensitive receptors, and sense any change of blood pressure and send signals to the higher centers, specifically vasomotor center, through these glossopharyngeal and vagus nerves. So basically, these sensory areas control the vasoconstrictor and vasodilator area and causes vasoconstriction and vasodilation for control of blood pressure. Vasomotor center also receives impulses from other structures like aortic and carotid baroreceptors, cardiopulmonary baroreceptors, carotid and aortic chemoreceptors, various ascending and pain pathways. Vagus lungs also sends signals through the vagus nerve, limbic cortex via the hypothalamus. So these are the various areas which send signals to the vasomotor center. So in case of neural mechanism, first we studied about the various areas which are responsible for the regulation of blood pressure. Now come to the mechanism through which these control are done. The important one is the autonomic regulation. Autonomic control of blood pressure, it is mainly done by the sympathetic control, because the systemic blood vessels are mainly innervated by the sympathetic fibers. They lack parasympathetic innervation. So in case of blood vessels, there is no innervation by the parasympathetic system. There are no parasympathetic innervations in case of blood vessels. They are only having a supply of sympathetic fibers. In this note, we have mentioned that these systemic blood vessels are not supplied by the parasympathetic fibers. Sometimes vagal stimulation also decreases blood pressure by decreasing the heart rate and cardiac output, but directly they are not having any nerve supply through the parasympathetic fibers. So there are two systems of sympathetic which is responsible for regulation of blood pressure: the sympathetic vasoconstrictor system and the sympathetic vasodilator system. Both the innervations are done by the sympathetic fibers. These are the structures which are involved in autonomic regulation. Now, the sympathetic vasoconstrictor system. Basically, there are noradrenergic fibers which supply blood vessels and responsible for vasoconstriction. So stimulation of these fibers finally results into the increase in blood pressure. So there is the increase in blood pressure when these noradrenergic fibers got stimulated, and they are responsible for the vasoconstriction. Sympathetic vasoconstrictive fibers basically have a basal rate of tonic discharge, which is important to maintain the normal vasculature tone. Such kind of basal rate of tonic discharge is not present in case of parasympathetic, sympathetic vasodilator fibers. Sympathetic stimulation increases blood pressure through these mechanisms. The important one is the vasoconstriction. Second one is the venoconstriction. Cardiac stimulation increases heart rate and myocardial contractility, which finally results into the increase in cardiac output. If there is an increased secretion of catecholamines, mainly dopamine, adrenaline, noradrenaline, they cause uh, increase in blood pressure. Now come to the sympathetic vasodilator system. Arteries and arterioles of skeletal muscles are innervated by the sympathetic cholinergic fibers, which are vasodilator in nature. So they causes vasodilation and decrease in blood pressure. But these innervations does not have any kind of basal tonic discharge. Such kind of basal tonic discharge was present in case of sympathetic vasoconstrictor system, because that basic contractions or basic tone of vasoconstriction is uh, important for normal perfusion of tissues through the blood. Till now, we have discussed about the various centers which are involved in short-term regulation and various mechanisms through which blood pressure is controlled, mainly vasoconstriction and vasodilation. Basically, there are sympathetic innervations which have both vasoconstriction and vasodilation. In case of blood vessels, there is no nerve supply through the parasympathetic nerve fibers. Both vasodilation and vasoconstriction is done by the sympathetic nerve fibers. The next section in case of neural mechanism is the reflex regulation. Various reflexes we are going to discuss through which blood pressure could be controlled. The important reflexes are the baroreceptors reflex, chemoreceptor reflex, Cushing's reflex, and Bainbridge reflex. The first and the important one is the baroreceptor reflex. It is also called sino-aortic reflex. So basically, high blood pressure or high pressure baroreceptors are located in the ventricles and the arterial side of the circulation, and the low pressure baroreceptors are mainly present in case of atria and pulmonary circulation. This is the whole uh, network which is responsible for the baroreceptor reflex. Now, what is the physiological significance of baroreceptor reflex? If we are having a very basic example, if a person is lying or sitting in a relaxed position and suddenly he is standing. So in case of this, almost 30%, uh, there is almost 30% reduction in blood pressure. So this reduction could results into shock kind of conditions. But in general, it doesn't happen because baroreceptors got activated, and these baroreceptors are responsible for the regulation of blood pressure in a very short span. Almost it requires 15 to 30 seconds for activation of these baroreceptors to regulate blood pressure. How baroreceptor sense blood pressure. Basically, if there is rise in arterial pressure, it stretches carotid baroreceptors, which transmit signals through Herding's nerves and glossopharyngeal nerves to the nucleus tractus solitarius. And this nucleus tractus solitarius finally sends signals to the vasoconstrictor and vasodilator area for further regulation of blood pressure. In case of aortic baroreceptors, which are present in the arch of aorta, signals are transmitted through the vagus nerves to the nucleus tractus solitarius. In case of carotid baroreceptors, Hering nerves and glossopharyngeal nerve is responsible for sending signals to the NTS, and in case of aortic baroreceptors, vagus nerve sends signals to the nucleus tractus solitarius. Now, how the blood pressure is controlled through these baroreceptors? When there is an increase in blood pressure, baroreceptors got activated and they send stimulatory impulses to the nucleus tractus solitarius, which is present in the cardioinhibitory centers through the glossopharyngeal and vagus nerve. Now, this nucleus tractus solitarius sends signals to the vasoconstrictor area and vasodilator area, which are the areas of the vasomotor center, and both these mechanisms, both these areas are responsible for the regulation of blood pressure. It inhibits vasoconstrictor area and excites vasodilator area, because in initial the level of the blood pressure is increased. So by inhibiting vasoconstrictor area, vasoconstriction is decreased and it causes decrease in blood pressure, and there is an stimulation of vasodilator area which causes vasodilation, and finally it's also responsible for the decrease in blood pressure. Same mechanism is done in case of vice versa condition, when there is initially there is a decreased blood pressure. In this image, we are having a flow chart, like the first condition in which there is an increased blood pressure. So by increased blood pressure, there is a stretching of these baroreceptors, and this sends signals through 9th and 10th cranial nerves to the nucleus tractus solitarius, and finally this nucleus tractus solitarius sends signals to the vasomotor center, where we are having vasoconstrictor and vasodilator area, and it controls or decreases blood pressure by inhibiting vasoconstrictor area and increasing or stimulating vasodilator area. Same condition or the same mechanism is followed, but in opposite direction. In case of when there is a decrease, initially there is a decreased blood pressure. The next mechanisms are the chemoreceptor reflex. So basically, these chemoreceptors are located in case of aortic and carotid bodies. Chemoreceptors, as their name suggests, they sense any kind of change in chemical composition, mainly hypoxia, hypercapnia, and acidosis. These are some specific conditions which could be sensed by these chemoreceptors. Whenever blood pressure decreases, blood flow to the chemoreceptors also decreases, which results in decreased oxygen content and excess of carbon dioxide, which is the hypoxia and hypercapnia condition, and it could be sensed by these chemoreceptors, and these chemoreceptors finally sends signals to the vasoconstrictor center, and finally this vasoconstrictor area or center causes vasoconstriction, and finally there is a increased blood pressure. These are the carotid bodies where we are having chemoreceptors. Aortic bodies, they send signals to these higher centers, and through various mechanisms like vasoconstriction. Some catecholamines are also responsible in case of control of blood pressure through the chemoreceptors.